Good morning, everyone. Welcome. Our speaker today is Dr. Sarah Drawz. She is a current resident here. She comes to us from Case Western University, and next year will be a hematopathology fellow here. And today is talking about antibiotic resistance. Take us away.
Thanks, Evan. So I'm going to talk today about antibiotic resistance, which is a topic we don't discuss a lot in this format of pathology grand rounds, but is something I've thought a lot about. My PhD thesis work was looking at the mechanisms of resistance to beta-lactam antibiotics in gram-negative bacteria, specifically E. coli, Pseudomonas, and Klebsiella pneumonia. So when I first came to the university 4 years ago to start residency, I was interested in continuing that type of work, and I was fortunate enough to get put in touch with Dr. James Johnson, who is a clinical infectious disease doctor at the VA here, who has done a lot of work looking at resistance patterns in E. coli. So with his help, as well as Dr. Ferries from our department, and also Dr. Susan Klein, who's another clinical infectious disease doctor here at the university's department, we performed this study where we looked at the resistance patterns in our local E. coli populations that I'll be telling you about today. I have no disclosures. So over the next 40 to 45 minutes, I'm going to generally review the challenge of antibiotic resistance, and then I'm going to introduce this resistant E. coli clone called ST131. I'm going to explain why it's relevant to the healthcare system today, to patient care, and why I think it's relevant to the laboratory. And I'll spend really the first half of my talk giving this background on the epidemiology and resistance patterns of ST131. And then in the second half of the talk, I'm going to review our specific study findings and what it taught us about our local E. coli population. So I'd like to start with some general perspective. Microbial infections are a significant drain on our healthcare resources and also a threat to patient healthcare. It's been estimated that 5 to 15% of all patients admitted to the hospital will acquire a microbial infection while they're in the hospital. So this would qualify as a hospital-acquired infection. And this is for people who come in with any diagnosis. Regardless of why they came in, they'll get an infection while they're here. And as you may be able to surmise, the people who are in the ICU are often the more compromised patients. They have multiple comorbidities that might make them more susceptible to infections. And in fact, they are much more likely to get these hospital-acquired infections. And if you look at Medicare and Medicaid data over the last 10 to 15 years, it's estimated that the average cost of a hospital stay for a patient without the complications of an infection is about $11,000. So this is all comers, all diagnoses, all lengths of stay, just to get some perspective. Because when we look at the average cost of a hospital stay for a patient with a hospital-acquired infection, those studies have estimated the cost median to be about $150,000. So you can see there's a significant increase in cost. And a drain to our already stressed healthcare system, it's been estimated it costs about $40 billion a year to cover these hospital-acquired infections. So fortunately, we have a set of pharmaceutical agents that help us combat most of these microbial infections. Arguably, the discovery of penicillin in the late 1930s and its development in the 1940s is one of the most significant advances in modern medicine. And antibiotics, as a general group, work by inhibiting different processes in cellular metabolism and synthesis. So this cartoon represents a bacterial cell, and then we have the different broad groups of antibiotics around the border and how they inhibit cellular processes. So nucleic acid synthesis inhibitors, protein synthesis inhibitors, and then also cell wall synthesis inhibitors. And I'll be talking about resistance in general to antibiotics, but I'm going to focus specifically on a DNA gyrase inhibitor, a fluoroquinolone ciprofloxacin, and then also resistance in this E. coli clone to one of the cephalosporins, or rather to several of the cephalosporins, which are members of the beta-lactam group of antibiotics, which work by inhibiting cell wall synthesis. So unfortunately, antibiotics don't always work. We're well aware of this as members of the medical profession. It's also become pretty high attention in the public media as well. It's been estimated about 70% of bacteria are resistant to at least one antibiotic. This translates to about 2 million infections with antibiotic-resistant bacteria a year in the US and about 90,000 deaths. In 2004, the Infectious Disease Society of America did a policy report called Bad Bugs, No Drugs, drawing attention to this problem. And their subheader for the report, I think, nicely summarizes their main point, and that was, as antibiotic discovery stagnates, a public health crisis brews. This is nicely illustrated if we look at the rate of development of new antibiotics over the past couple decades. This graph on the right shows that we've had a significant decrease in the release of new antibiotic agents over the last several decades, and we've really reached our nadir here from 2008 to 2012, when only 2 new antibiotics have been introduced into the market. And if we were to look at a comparable graph over the same period of time for how organisms are developing resistance to antibiotics, we would see, of course, that it goes in exactly the other direction with a pretty rapid increase. So in 2010, the Infectious Disease Society of America introduced another policy report and campaign to try and conjure up some energy and effort to discover and introduce new antibiotics as a collaboration with the legislature, pharmaceutical agencies, as well as research. And this new policy and movement was called Bad Bugs Need Drugs, a lot of catchy phrases from the IDSA. And their main point or goal, or rather I guess challenge really to the development of antibiotics, was to introduce 10 new antibiotic agents by 2020. So that's that 10 by 20 pill here. And this pictorial graphic shows our progress. So since 2010, there's just been 1 new antibiotic introduced into the market. So we are significantly behind pace if we want to actually introduce 10 new antibiotics by 2020. So what can we do about this problem of increasing antibiotic resistance and the decrease in discovery of new drugs to help control these infections? It's a focus of a lot of research in infectious disease in particular. Some of the questions that we try and answer are why the current antibiotics that are being used or misused select for particular resistant strains. What is it about these specific resistant strains that makes them more likely to resist the antibiotic pressures and achieve dominance? Is it that these resistant organisms are necessarily more virulent than other non-resistant counterparts? Or are they less virulent in particular ways? Or does it fall more in the lap of the healthcare providers? Is it because we aren't practicing infection control like we should be to control the spread of these organisms? Now, of course, most likely it's a combination of all of these factors, but really trying to focus in on which one of these we have the most control over and which are the most effective for controlling particular antibiotic-resistant organisms is the focus of this type of research. And so our study in particular looked at this clone of E. coli that's particularly resistant. It's also been, as coined by Dr. Johnson, sweeping the world. It's called sequence type ST131. This is a recent review that Dr. Johnson published with one of his collaborators, Dr. Banerjee from Mayo. So I want to start with, give you just a little background on E. coli as an organism. We're probably all pretty familiar with it, but it is a gram-negative bacteria and generally lives as a commensal organism in the gastrointestinal tract of both humans and animals. And the vast majority of the time it lives there just fine. It's in a harmless symbiotic relationship with our other gut flora, helps us digest some of our dietary sugars and benefit both ways. But of course, a subset of E. coli is pathogenic. E. coli as a group is the leading cause of urinary tract infections, as well as soft tissue infections and bloodstream infections. And E. coli as a group has developed resistance to every different class of antibiotics. Not necessarily one clone, but different types of E. coli have acquired this resistance. And it has been noted that E. coli as a group has a significantly rising resistance pattern in the recent 10 to 15 years. So what is this ST131 I keep talking about? So ST stands for sequence type. So this is a particular way to describe genetically strains of E. coli. The sequence typing is done by genotyping 7 different housekeeping genes. Most of these housekeeping genes are related to sugar metabolism and also DNA synthesis. So there's a whole series of different ST numbers. But our organism of interest is ST131. It's also a member of phylogenetic group B2. So E. coli is divided up into phylogenetic groups based on their evolutionary relatedness. So for example, phylogenetic group A is generally the commensal E. coli, whereas those that are in group B or B2 or even D are usually the pathogenic E. coli. And E. coli ST131 is also a member of serotype O25H4. The majority of them are. Some of them are O16. As a reminder, this OH designation refers to the O antigen, which is a component of the lipopolysaccharide cell wall layer, and then the H refers to what type of flagellin that particular E. coli possesses. So SC131 was first described in 2008, and it was described by a group of researchers who were studying this increasing resistance in E. coli organism. In particular, they were looking at E. coli that were novelly expressing an extended spectrum beta-lactamase called CTXM. So beta-lactamases are enzymes that hydrolyze beta-lactam antibiotics. So as you may recall from the earlier slide, beta-lactam antibiotics include penicillins. They also include cephalosporins, carbapenems, and aztreonam. So extended spectrum beta-lactamase is an enzyme that can hydrolyze the extended spectrum beta-lactams. And that includes extended spectrum cephalosporins such as cefotaxime, ceftazidime, ceftriaxone. So this particular enzyme, CTXM, is extremely efficient at hydrolyzing cefotaxime in the bacteria that express it. It's actually named CTXM because of an acronym for cefotaxime. So they were studying this E. coli that had this increasing resistance pattern. It harbored the CTXM, and they were surprised to find that there was a similar clone, this ST131, in 9 different countries on 3 continents, in North America, Europe, and Asia. They further described that this strain of E. coli was a member of the XPEC E. coli group. This is another acronym, which stands for extraintestinal pathogenic E. coli. So these E. coli have acquired the ability to cause infection outside of the gastrointestinal tract, as the name implies. They're particularly efficient and well known for causing bacteremias, intra-abdominal infections, pneumonia, as well as wound infections. And SC131 in particular has recently become the most common strain of E. coli causing sepsis. And as you can see, that carries a pretty high mortality rate. And SC131 is also, as I have alluded to, resistant to multiple antibiotics. There's some subclones which are particularly resistant that I will discuss in future slides. But as a group, SC131 is generally resistant to the penicillins, aminoglycosides like gentamicin, as well as Bactrim. So this slide, I think, has a nice visual representation of the resistance patterns that we see in E. coli ST131. So first I'm showing you a Kirby-Bauer disk diffusion assay for a non-ST131 E. coli. So most of you are probably familiar with this disk diffusion assay, which is used in the microbiological laboratory to determine the resistance patterns of a particular organism to antibiotics. So it works by plating a particular pathogen on this agar plate, and then also on this agar plate are these paper wafers that are impregnated with antibiotics. And so when they're placed on that plate, the antibiotic substance diffuses into the surrounding agar, and if the pathogen that's on the plate is susceptible to that particular antibiotic, it won't grow in that area. And so this clearing is called a zone of inhibition. And this zone of inhibition can be calculated into minimum inhibitory concentrations, which are, of course, the values that the laboratory releases to clinicians to help inform the treatment of resistant or non-resistant infections. And so we can see that with a non-ST131 E. coli, it is susceptible to all of the antibiotics at different levels that are on this plate, with the exception of this one. But if we look at a similar test for an ST131 subclone called H30RX, which is particularly resistant, again, I'm not focusing on which particular antibiotics it is resistant or susceptible to, but I hope you can appreciate from this visual image that it is much less susceptible to the majority of these antibiotics. In fact, it really isn't even touched by those that had a really pretty significant zone of clearing on the non-ST131 E. coli plate. So ST131 not just very resistant, but also extremely efficient at spreading throughout the world. So it has reached pandemic proportions in the past 10 years. As coined by these authors of clinical microbiology reviews in the last year, it is spreading explosively throughout the world. This is a map of the world from their review, and the blue and red stars represent the different areas that have described a significant population of ST131 in their E. coli population. And so you can see that from its rather meager beginnings in 9 countries on 3 continents, it's now pretty much all over the world. The prevalence of ST131 as a member of the general group E. coli varies depending on which region of the country you're in or what patient population you're looking at, but it's generally about 13 to 30% of all E. coli. And it is thought that the presence of this ST131 subclone is really driving the increasing resistance that we're seeing in E. coli as a group. So I've been talking about ST131 and then alluded to some subclones. ST131 is not a single unitary entity, but it is composed of several subclones. This was recently elucidated by some whole genome and single nucleotide polymorphism sequencing studies that looked at the phylogeny of ST131, and what they found was that there were nested subclones of ST131, which interestingly were all derived from a common ciprofloxacin susceptible ancestor, and that these subclones represented a resistance continuum. So this figure represents the phylogenetic tree of ST131, and we start here on the left with this common ST131 ciprofloxacin susceptible ancestor, and then a subclone of this group broke off and acquired this fimbrial H30 gene. It was denoted H30. And then a subclone of that broke off, acquired resistance to ciprofloxacin, referred to here as fluoroquinolone. And then a subgroup of that, H30RX, acquired this enzyme for CTXM and resistance to the extended spectrum beta-lactamases. And so this is particularly interesting because oftentimes when you have resistant clones popping up from in any particular group of bacteria, you have independent emergence of those resistant clones because of different episodes of antibiotic pressure, whereas here we have them all from one common ancestor, and each one of them are successively more resistant. So why is ST131 so successful at spreading the way it has? We really don't understand that very well, and if we want to help control the spread, certainly it would be good to have a better understanding of why it's so able to sweep the nation in the way that it has, or rather sweep the world. So there are several hypotheses as to why ST131 has been so successful in its pandemic, and there's varying levels of data to support each of these hypotheses. One of the most obvious is that ST131 is just really resistant. So because it's so resistant and because we use and misuse so many antibiotics, the selective pressure is perpetuating the dominance of ST131. But even if that's true, we could also ask, well, then why is ST131 taking over? There's other strains of resistant E. coli. What is it about this particular one that makes it so successful? What about transmission? There have been some case reports documenting person-to-person, really efficient person-to-person transmission of ST131, both within families, members living in the same home, within daycare centers, within long-term care facilities. So it may be that ST131 is just really able to jump from person to person, and that's why it spreads like it does. It could also be that it's a really successful colonizer. It's been theorized that having ST131 in your gut, even as a healthy carrier, is a prerequisite to getting the infection. So maybe ST131 is really able to adhere to the mucosal surface and just lingers waiting for its opportunity. It may also be that ST131 is more virulent or more pathogenic than other non-ST131 E. coli's. It may be more fit in its particular ecological niche. There's been a series of epidemiologic studies suggesting that perhaps it is more virulent. It seems better able to form a biofilm than other strains of E. coli, and of course forming a biofilm is an effective way to resist antibiotic penetrance. And it's also particularly able to adhere with lots of different adherence virulence genes, so maybe that's why it's able to spread. But there's conflicting data from the animal studies of ST131 where it actually is shown not to be more virulent, so we don't really understand this very well. And counterintuitively, it may be that ST131 is actually less virulent than the non-ST131 counterparts. And this harkens to a paradigm that's familiar in microbiology, and that's that whenever an organism requires resistance, that resistance comes at a cost and becomes less fit in some way. I think a nice illustration of this is the resistance of Mycobacterium tuberculosis to rifampicin. So rifampicin works by inhibiting the DNA-dependent RNA polymerase in Mycobacterium tuberculosis. So when the organism requires resistance to rifampicin, it's because of mutations in that RNA polymerase. So while that helps it escape antibiotic pressure, you can imagine that it's probably less efficient at transcription because of those mutations in the RNA polymerase. So perhaps ST131 is actually less virulent than its other counterparts, and perhaps its resistance comes at a cost, which might be an opportunity for antimicrobial therapy if we can sort of nail in and hone in what that virulence cost is. So what did we do? So we wanted to look at ST131 in our local hospital system in Minneapolis. So we sought to determine the prevalence of ST131 and its subclones. We wanted to compare ST131 and its subclones with the other E. coli in our hospital system to look at virulence factors as well as resistance phenotypes and genotypes. And we also wanted to determine whether or not the E. coli, in particular the ST131 E. coli, was part of the global expansion that's been described, or maybe we just have our own local clonal outbreak. So this is a screenshot of the paper that we put together with all of our results. It was just finally published in press last month. I obviously owe a great debt to all of these co-authors, and I'll thank them more individually later. But all the data that I'll be showing you in the subsequent slides comes from this particular paper. So our methods were that we collected all clinical E. coli isolates submitted to the Infectious Disease and Diagnostic Laboratory over a 7-week period. So as many of you may be aware, the Infectious Disease Diagnostic Laboratory, located on the 2nd floor of Mayo under the direction of Dr. Frary and Dr. Arbefeville, processes all microbiological specimens from Masonic Children's Hospital, the East Bank Hospital Facility, and the West Bank Hospital Facility. So all of those come through the Infectious Disease Diagnostic Laboratory, which is a great opportunity for us to sort of capture all of those specimens in one place. And interestingly, and as many of us are very familiar, these different hospital systems treat very different patient populations. So the Children's Hospital, as the name suggests, treats just children. The East Bank Hospital has a range of very acute and complicated diagnoses, many transplant patients, both solid organ and hematologic, as well as immune-compromised and oncology patients. And this is in rather stark contrast to the West Bank, which has services mostly focused on orthopedics, rehabilitation, obstetrics, and psychiatry. However, each one of the facilities does have an emergency department and an intensive care unit. So while there's a spectrum of care and patient population in each one of the hospitals, we did think this was a unique opportunity to study different types of patient populations within a geographic locale. So again, all these specimens came through the IDDL. And in the IDDL, once an organism was identified as E. coli, it was then moved to the antibiotic susceptibility desk, where the medical technician sitting on that desk for the day took the extra time and energy to plate that E. coli specimen on a plate just for me for this study, which I appreciated. I would go pick up periodically these plates of trays that they had made and incubated for me. I would record down where the specimen had come from, which hospital, where in the body, was it from wound, urine, soft tissue, and I then would also record its ciprofloxacin and extended spectrum beta-lactamase status, as was determined by the Vitek instrument. I then moved it to specimen management, where the people there helped me package it appropriately for infectious and hazardous material. I then went off to the Minneapolis VA, where Dr. Johnson's laboratory took over. And Dr. Johnson's laboratory scientist did a lot of phenotypic description of the E. coli isolates, which I was able to help with. We looked at phylogenetic group for each one of the strains that we isolated. So again, these lettered groups, there's a total of A through F. We also figured out what its sequence type was. Was it ST131 or one of the other sequence types? We then looked for the presence of 50 different virulence genes, and we confirmed the presence of the CTXM beta-lactamase. And we ended up with a lot of data, a lot of epidemiologic data, a lot of phenotypic data, with 6 total tables. So I'm not going to go through all of that today. I'm just going to focus on one part of the story, in particular this ST131 and its subclones. So you'll see my little summarized tables here. So this is essentially a summary of Table 1. So this describes the hospital of origin as compared to resistance status, and then I'll also show you for lineage groups. So across the top, we can see our general numbers that we collected a total of 233 specimens over the 7-week period. The vast majority came from the East Bank. Again, these are all the E. coli's, but we certainly still had a contribution from the West Bank and the Children's Facility. And if we look at those isolates that were ciprofloxacin resistant or had an ESBL present, we can see that there is a significant association between hospital of origin and both of these resistance determinants. So these numbers represent the number of isolates, so 65 total ciprofloxacin resistant isolates, a total of 28% of the 233. So the percent found in the parentheses represents the percent for that column. So we found, as these first 2 lines are showing, that ciprofloxacin resistant and ESBL positivity were associated with hospital of origin. And if we look then just at phylogenetic group B2, again, we determined it for the others, but I'm not showing it here. we see that group B2 was the most predominant phylogenetic group that we found in all of our 233 specimens. So ST131 is a member of group B2. And if we do our subgroup analysis and look at just ST131, as compared to all the other serotypes that we did identify and collect, ST131 was the dominant lineage that we saw in all three of these hospitals. Now, interestingly, it had rather similar prevalence across the different hospitals. So about 22 to 27% of the total isolates from each of these facilities were ST131. So, of course, while the majority of the ST131 did come from the East Bank, so did the majority of our specimens. But interestingly, the total percent prevalence of ST131 as a proportion of the E. coli population at each of those sites was comparable. And we can then look at the subclones H30 and H30RX. Again, H30 is the one that broke off and acquired resistance to fluoroquinolones. H30RX has got both the fluoroquinolone resistance as well as cefotaxime resistance. And these two subclones were predominantly seen at the East Bank and interestingly not seen in any of the Children's Hospital isolates. They did exist in a gradient from East Bank to Children's, and it was significantly associated with hospital of origin for the H30 subclone. On the second table, we're looking at the relationship of phylogenetic status, and then we'll also look at clonal group as compared to resistance determinants, both for ciprofloxacin susceptibility and resistance, and ESBL negativity and positivity. Now, again, this represents the presence of that CTXM extended spectrum beta-lactamase. So if we look just first at these different phylogenetic groups, we see that generally, with just one exception, there is no significant association between the phylogenetic groups and the resistance determinants, which is somewhat interesting for group B2 because we know that it harbors our ST131 subclone. Although when we do the subgroup analysis, we do see that ST131 and its subclones are the most significant and predominant carriers of both ciprofloxacin resistance and ESBL positivity, with some very significant associations. So even though B2 as a general group was not associated, our resistance subclones are. So how about our virulence studies? Again, we looked at 50 different virulence genes, and after determining the genetic status of those 50 different genes, we then calculated an aggregate score for each organism, and then as a group of organisms. So here we are looking at the aggregate virulence score median for the subgroups of ST131, H30, and H30RX. And if we compare the median virulence score, so in other words, an estimate of how virulent that particular group is with this scoring mechanism, if we compare that score to all other E. coli in our study, we interestingly see that ST131, by this virulence score and its subclones, is not really more virulent than all other E. coli. This was similar to what we found when we looked at ciprofloxacin susceptible subgroups of our population and ESBL negative subgroups of our population. So in other words, when we looked at those groups, they were not necessarily more virulent than their non-ST131 counterparts. This changed when we did the subgroup analysis with the resistant subsets of the population. So here I'm showing you the ST131, H30, and H30RX, the resistant ones, which really includes all of them, compared to the virulence scores of all the other resistant E. coli in our study. And same thing with ESBL positivity. And we can see that overall, now with the subgroup analysis, when we're looking at just the resistant strains, that ST131, H30, and H30RX do have significantly higher virulence scores than their resistant counterparts. Okay, so thus far, we showed that ST131 is the dominant clone in our local population, has a comparable prevalence across our different hospital sites. ST131 and its subclones are the main carriers, or ciprofloxacin resistance, and the ESBL CTXM. And then among just our resistant isolates, ST131 and its subclones have higher aggregate virulence scores. So this is how we submitted the paper originally with compilations of this data. And one of the reviewers brought up a relevant and important question, and that was, Are the authors sure that the high prevalence of ST131 subclones H30 and H30RX in the East Bank facility is not due to the spread of the same strain in this facility? So in other words, are we bringing in multiple strains of ST131 from the community, or do we just have a local clonal outbreak at our East Bank facility? So they recommended future studies, in particular pulse field gel electrophoresis, to try and determine whether or not this had just been patient-to-patient transmission in the same facility. So like you do when a reviewer brings up a good point and suggests more studies, you do them. So we did PFGE, which is, again, pulse field gel electrophoresis, which is a molecular typing technique to look at bacterial fingerprints. So essentially from this type of study, you can determine with a good degree of certainty whether or not different bacterial isolates are the result of one clonal, or rather not clonal, but local strain outbreak. So it's performed by starting with your bacterial culture. You take bacterial cells, you then embed them in an agarose matrix, and then release the DNA so that you can cut it up with characteristic restriction enzymes. And then it is then placed in an electrophoresis cell, and electrical currents are applied from different directions over a long period of time, typically at least overnight. And this essentially allows these fragments of DNA to wiggle their way through the agarose matrix in characteristic patterns depending on how big they are. And in the end, you end up with this bacterial fingerprint, which is essentially just your different bacterial isolates run through the gel, and the bands represent the fragments of DNA at different locations based on its particular genotype. And this can be developed with UV light. So PFGE end result is a dendrogram, and this is our dendrogram, so it represents how related our different isolates were. So we performed this PFGE for all of our ST131 isolates, about 51 of them, 61 rather. And this is the different rows, rather, of the gel that show the DNA fragmenting pattern. So these are essentially each little pictures that were taken of the gel with the UV development. And it is then inserted, this pattern can be scanned and inserted into online databases, which will then tell you how related is a pattern that looks like this at the top to a pattern that looks like this, and give you this phylogenetic bracketing on the left. And so organisms that are in brackets together are more related, and organisms that have shorter horizontal distance are more related. So in other words, these organisms right here at the top, the two are very closely related, but certainly not closely related to those towards the bottom of the graph. And so when we analyzed the different pulsotypes, as they're called, from our ST131 isolates, we found 38 different pulsotypes. And many of our more frequent pulsotypes were those that have been described in other studies of ST131 in the United States. So this suggests strongly that our ST131 population does reflect sort of the national ecology, and it's not just one clonal in one institution outbreak. So with that, we resubmitted the paper and it got accepted. And so our three primary findings are that of all the E. coli in our local population, ST131 is the dominant lineage group. We had a unique comparison of 3 different hospitals within the same geographic locale that had a spectrum of acuity in different patient populations. But interestingly, ST131 as a component of all the E. coli was similar across the hospitals, and also similar to those prevalence percents that have been found in other studies, in particular a study that Dr. Johnson's group did looking at United States veterans, and then also a study performed by one of his collaborators down at Mayo. Our second main finding was that ST131 and its subclones are the main carriers of ciprofloxacin resistance and the ESBL CTXM. And that when we look at aggregate virulence scores, they're comparable for ST131 and all other E. coli, but when we look just at the resistant phenotypes, ST131 does appear to be more virulent. So what does this all mean, and what can we do with it and learn from it? Well, I would argue that sort of our most significant summary is that ST131 comprises a significant population of both at least relatively virulent and resistant E. coli isolates in our Midwestern locale. And as I showed earlier, we know that ST131 isn't a single unity. It's rather composed of these multiple nested subclones, and our study was one of the first to really look at those nested subclones and to try and determine what their epidemiological correlates are. In other words, who's more likely to get one of those subclones? What is it about the patient or about the facility that puts them at higher risk? And so we, in our study, really used hospital as a surrogate for ecological context, which we arguably can do, at least to some degree, because of the different patient populations that are seen in the East Bank, West Bank, and the children's facility. And so it does appear that at least those hospital contexts do impact what the clonal mix of the E. coli is in that particular group of patients. In other words, we have this negative association between the children's hospital and the resistant clones H30 and H30RX. This negative association has been described in part before for children at least ages 11 to 20, typically don't have H30 or H30RX. They have other ST131s. Not entirely clear why that is. Certainly children are exposed to probably different antibiotic patterns. They have different comorbidities. But does that inform how at risk they are for those subclones? We're not really sure. And we also showed a pretty strong positive association between the East Bank and the resistant H30 and H30RX subclones. So again, the East Bank patients, they're severely ill. Oftentimes they're immune compromised from transplant. They're often on multiple antibiotics. So it may be that something about that niche really favors antibiotic-resistant pathogens. So I think about it this way. We have our 3 hospitals with our 3 different patient populations, and ST131 as a component of the E. coli populations at these 3 facilities is comparable percent-wise across these 3 facilities. But when we look at H30 and H30RX, we don't see these resistant subclones at the Children's Hospital. They're still present at the West Bank, but at lower numbers than they are in the East Bank. So what is it about this East Bank facility or the patient population that can help us understand that? And then when we process our virulence results, again, our ST131 was not more virulent than all other E. coli, but with our resistant subsets, it was significantly more virulent. So we can interpret this as ST131 may not be overall more virulent. Perhaps it's really the strongest and the fittest of the resistant, so sort of its subgroup, and that this relative virulence has promoted its rise in the population. So this, would sort of challenge this general paradigm that ST131 as a resistant organism has some significant cost to its resistance, but perhaps it actually has a little bit lower cost to its resistance because it is able to remain more virulent than its resistant counterparts, which under a great deal of antibiotic pressure is really its main competitors, so to speak. So overall, and in summary, our Midwestern locale is part of the ST131 global pandemic. Our PFGE results suggest strongly that we are part of the global outbreak and not just reflecting a local clonal outbreak within our community. The ST131 subclones have distinct ecologic, epidemiologic, and clinical associations as inferred from the different hospital populations. And that ST131 does appear to be both resistant and relatively fit. So now what? Moving sort of forward and building on these results, I would argue that based on what we found here, that H30 and H30RX, less prevalent in children, what is it, again, I alluded to this, about children that makes them less at risk for H30 and H30RX? Or is it just that we didn't get enough isolates from the children's facility to pick that up because E. coli, ST131, and E. coli in general is not as common in that patient population? It could be independent of children. There's probably a lot of other patient-level risk factors that we don't entirely understand that put people at risk for these particularly resistant infections. Is it the particular antibiotic regimens that they've been on? Maybe invasive devices? Is it residents at long-term care facilities or other facilities that may promote person-to-person transmission? Interestingly, some studies of ST131 have shown that international travel puts people at risk for these infections, in particular travel to India. So clearly a lot of additional work needed to really understand why the spread has occurred and what we can do to sort of stop it, and also patient outcomes in these particular subsets of people who get the ST131, H30, and H30RX infections. Will the pandemic be helped by improved antibiotic stewardship or infection control? These are also important questions to keep in mind. Certainly antibiotic stewardship has an important place. Misuse of antibiotics is something that will probably benefit the resistance profiles in general, as is infection control. But interestingly, specifically for the ST131 clone, we don't actually have any really good data to suggest that either one of these approaches will help control the spread of ST131. So a lot of opportunity for documentation in that regard. So I would argue that when we still don't really understand what the good clinical predictors are of patients that are going to get these types of infections, that it underscores the role of the laboratory in identifying these organisms, communication between the laboratory and clinicians certainly for when these particularly resistant strains of E. coli are coming through and being isolated and tested. And there's also been some discussion that in sort of our perfect world that there would be rapid diagnostics that would help us pick up these particularly resistant subsets of E. coli more quickly. In other words, perhaps before antibiotic susceptibility testing had been turned around, because that often requires growth. There's been some discussion that next generation sequencing perhaps could be applied to identification, as well as mass spec MALDI, which is MALDI is a technique currently being used in our microbiology laboratory. And identification of ST131 as a subgroup of E. coli is something that has been explored from a research basis. In particular, a recent paper looked at ST131 and also other STs of E. coli. And this is just a screenshot of one of their images, one of their spectra, where they looked at different E. coli strains. And you can see that the black peaks represent that proteomic profile on MALDI for specific ST131, whereas the other colors are the other strains. So perhaps there's some future application using using MALDI to help sort of get a jump start on whether or not a particular infection might be caused by one of these resistant strains. So I want to be sure to thank Dr. Ferreri, Dr. Johnson, and Dr. Klein for helping make this project possible and helping me with all of the logistics as well as the interpretation and write-up. In particular, also to thank the Infectious Disease Diagnostic Laboratory. It was most often David Goosey who was sitting on the antibiotic susceptibility desk who had to plate those extra E. coli strains for me, so thank you to him. And then the individuals in Dr. Johnson's lab at the VA who did so much of that PCR characterization of the strains. And then Mike Kukowski has helped us with our statistical analysis. And I just have one more thing to point out before I take any questions, and that is the significance of today's date. So today is October 21st, 2015, and I have Dr. Sarah Williams to thank for bringing this to my attention, because not only is this her 4th wedding anniversary, but today is also Marty McFly Back to the Future Day. So many of you may remember these really popular movies in the 1980s, Back to the Future and Back to the Future 2, and Back to the Future 2, they come forward 30 years to save Marty's yet-to-be-born children from some certain despair. And they set their future time machine to October 21st, 2015. So we are now here, and there's a lot of discussion on the popular media about how to celebrate and what's going to happen today. There's been countdowns online, and so this is actually from one of those websites. Is today Back to the Future Day? And you would put in whatever today's date was and say, nope, it's not October 21st yet. So today, this webpage is finally saying, yes, it's October 21st. And it's funny to think about what they thought 2015 would look like. We thought we'd be traveling mostly by hoverboards, we'd have flying cars, and we'd have a really cheap source of fuel. We'd be burning our garbage, which unfortunately is not true. Although, in the movie, the Cubs won the World Series in 2015. So, despite their rather insurmountable deficit right now, they may still pull it out. So that's all I've got. Dr. McKenna?
Sarah, and you may have said this and I might have missed it, but were all of these patients from which the organism was isolated clinically infected with the organism, or was it just cultured?
Yeah.
Were they carriers?
I didn't say that, and we did not collect that information. So that is something certainly we could go back and look at, but we, fortunately or unfortunately, didn't collect any specific patient characteristics. So we don't know. I mean, they were cultured, obviously, because that's how we ended up with them, but we don't know if they were clinically infected.
And these were all hospitalized individuals?
Yes, they could be outpatient, too. Outpatient or inpatient, yeah.
So, but is there data on the community as a whole, non-sick patients?
There isn't a lot of good data. There are a couple individual studies in particular trying to look at the healthy carrier rate, and there have been studies that have shown that all-comer healthy carrier rate is about 4%. There have been other studies that break it down, particularly by whether or not they harbor resistant strains in the general population, but we don't have a really good picture of what it looks like just walking around on the street. So Dr. Carson.
Oh, as a follow-up to Dr. McKenna's question, I do think that the clinical correlation here would be very important.
Yes, yeah, it would.
For example, if you have a specific virulence factor for maybe a virotoxin in some of these organisms, you might have patients who have a thrombotic microangiopathy, or
Sure.
you might be able to correlate some of the virulence factors with specific clinical
Yes.
findings.
Yeah, there really is not a lot known about that. Despite how widespread this strain is, we don't have a really good idea of what the clinical correlates are. And
Do any of these have a virotoxin?
we've looked at lots of toxins. There are. I mean, I'm not going to be able to tell you exactly which ones, but I can refer you to this figure in our paper. And these are all the different toxins that we screen for, if you can read this at all. There's adhesins, there's a series of toxins, there's siderophores, protectins, as well as other often metabolism toxins.
But many of them are urines, Sarah.
Many of them were urines, yes, yeah.
And so to answer Dr. McKenna's question regarding clinical correlation, they were sent in to rule out urinary tract infection, but we can't rule out that it could have been a fecal contamination of the urine rather than a true UTI.
True.
But the vast majority of the E. coli's were from urines, remember? We didn't include Fairview Ridges and Fairview
we didn't,
Southdale isolates. It was just the three that Sarah presented.
Dr. Hanson.
Hi, Sarah. That was really nice. A very nice
Thank you.
explanation of an increasingly complicated scenario. Just to follow up on the clinical correlation.
Yeah,
About
you
4
might
years
know.
ago, we sent all of our bloodstream infected E. coli isolates that we recovered from Hennepin to Scott Weissman in Seattle. And 90% of our E. coli
90.
strains that presented as bloodstream infections are ST1.
90, wow.
100%
That's-
of all of our ESBL producing E.
coli are
are
ST131.
So I do think, I agree
That's
with Dr.
great,
Freeman
yeah.
that that clinical data set is growing, and that's partly where some of that term XN comes from, that essentially infections that we see with E. coli that are non-GI related are a predominance of this individual clone. But I have I guess more of a thought for you than a question, because quite frankly, none of this makes any sense to me. But,
Awesome.
it
I
doesn't
did a
take
good job.
much these days. But everything we've known, you alluded to this, everything we've known about bacterial resistance and fitness suggests that the dogma is that resistance that organisms carry pay a price for that. And every study that's been done, going back to the early 2000s with Denny Stevens, who showed that acquisition of methylase genes in Staph aureus really hampered transmission and gave a proposed mechanism for the role of community-acquired Staph aureus, the so-called USA 300 strains, which almost unilaterally lack methylase, clindamycin resistance. And Tim Walsh has very strong evidence now that MDM1s are incredibly poorly fit relative to this. So I have maybe a thought I'd run by you. In the era of next-gen sequencing, you looked at a ton of genes, but I wonder if you applied next-gen sequencing to this type of research with control strains that were non-ST131 strains, if you specifically would find compensatory mutations. Because it doesn't make sense to me, your table. I mean, when I say that with love and affection.
Of course,
It doesn't
of
take
course.
much to confuse me, as usual. But you've got 4, 5, 6 virulence genes that you're detecting in these subclones that you're not seeing in the general population.
Right.
And theory would suggest that the price that we pay for carrying those is a decrease in fitness. It makes me wonder if we wouldn't see, if you targeted compensatory mutations in the genome using whole genome sequence technology, if you might see that come out, where then you could take the lineage of that research and restore that on plasmids in animal models to prove that we're really seeing increased fitness,
Right, because it
with
may not be.
associated virulence of these strains, because it's counterintuitive to almost, Dr. Ferrari's here, almost 40 years of bacteriology research. You've got a predominant clone that is sweeping the globe that has increased virulence, increased resistance, and presumably increased fitness.
Right.
It will be one of the biggest microbiological stories if that theory is proven.
As it actually plays out, It is, if there's anything I've learned in my relatively short career of studying antibiotic resistance, it's being humbled by how effectively, quickly, organisms acquire resistance. And I mean, their replication rate and the just baseline genetic mutation rate is just so high that I don't know how we're going to keep up.
Part of this research?
Well, I mean, there have been those whole genome studies, and I don't know whether specifically they've looked at the compensatory mechanisms that you're talking about. Yeah.
I was surprised, Sarah, that the reviewers did not ask whether the lab had studied expression of those virulence genes, because it's nice that he has the 50 gene virulence gene PCR assay, the multiplex, but studies that Jim has done in the past has looked at expression of a few selected virulence factors, hemolysin being one, and so on. And no one asked that in the reviews.
Right, just having that.
Responded to that if they had.
Well, certainly just having the gene doesn't necessarily mean that it confers any type of virulence. I mean, I think this would have been a little prohibitive in the scope of this study to try and look at each one of those virulence genes, but certainly we have to acknowledge that we're looking at it as just a marker, and we're inferring from just the presence of those genes that means that there's a cumulative virulence advantage. But I mean, you're right, it's not perfect.
Some organisms, there are pathogenicity islands that have a collection of virulence genes, and I don't know that data in E. coli.
I don't either. Yeah.
That's a good question.
Sarah, back to the PFGE experiment.
Yes.
You look at patients who came in with an infection versus those who acquired an infection within the hospital.
Yeah, so that would of course be very interesting, but since we didn't collect any of those patient level characteristics, we didn't even know which strains had come in or which strains had been transmitted theoretically in the facility. So we are not able to do that subgroup analysis. Although certainly someday maybe I could go back and collect all the patient level information on all of these people who contributed the E. coli strains, and that there would be a lot to learn from that. Yes.
You talked about this week it would roll, but in looking at the map, it would seem that there is less prevalence in the colder countries.
Yeah, Greenland hasn't reported recently.
And China. But is this just a factor that it has to be?
Yeah, and I mean, I do think so. There's just one red dot for China. I mean, I think that's sort of. And there's multiple dots for the U.S. I don't think this is meant to reflect the relative number of reports, but just that it has been documented in that area. And I don't think there have been any studies looking at sort of temperate versus non-temperate climates and whether or not that is a factor as well.
But they have very tough, there are parts of the world, New Zealand being one, Scandinavian countries, where they have very tough antibiotic stewardship rules. And they see very little methicillin-resistant Staph aureus, for example. They can count it on one hand usually. So they've been spared some of the problems that we experience in Western Europe, where we know the epidemiology, and in the United States in particular, and in South America, these resistance, and in India, are rampant. And so it may be that they have such tough hygiene rules also. Several years ago, one of our orthopedic surgeons went on sabbatical to a university in, I think it was Auckland, and he had to demonstrate that his nasal culture did not have plain Staph aureus in it. And so I personally did the cultures, oversaw the results, and wrote a letter that he was free of Staph aureus, and they allowed him to operate. But if he had been positive, he would not have been able to at the university in Auckland.
There is one study too, I can't remember if it's E. coli or KPC, where they sampled it. I can't remember the organism, but they sampled the latrine in airplanes.
Don't do that. Actually, it would probably be the counter that's worse. This is what they usually say. The toilet's okay, but the counter, the doorknob.
Don't you always wash those down, Jim?
Just don't sit. Yes, Dr. Cheese.
I have a question. What is known about animal reservoir? Because speaking about antibiotic stewardship, I think what we do in humans is nothing, and what is missing
Yes.
is stewardship in animal in farming.
Yeah. Well,
So could this similar study be performed in farming?
yes. So a couple thoughts on that. One is that the 2010 IDSA report that includes a lot of policy approach for trying to improve the problem of antibiotic resistance has a significant proportion of the report dedicated to the animal use of antibiotics, because arguably that's driving a lot of this. As far as ST131 in the animal population, there have been some surveillance studies, and it looks like that it is not very prevalent in animals. And so could you do this study in animals? Certainly, but the preliminary sort of epidemiologic screening studies suggest that it's not really predominant or even present at significant levels in animals, interestingly. Thanks.